Hi everyone, in this video, I'm going to present genome-wide identification and expression analysis of PUB genes in cotton. There are many proteins involved in cell cycle, stress resistance, and signal transduction. These proteins are modified by ubiquitin-mediated ubiquitination pathway which involve of three enzymes. First, ubiquitin activating enzyme, which activates ubiquitin. Activated ubiquitin molecules are then delivered to second enzyme, which is ubiquitin binding enzyme. Next, ubiquitin ligase is then used to transfer ubiquitin molecule to the target protein. In these three enzymes, ubiquitin ligase is much important for identification of the specific substrate protein. It contains of U-box subunit, which is encoded by plant U-box gene, PUB. Many studies state that PUB genes are involved in abiotic stress response. In this study, the main objective is using genome sequence, conduct a research for genome-wide identification, gene structure, evolutionary relationship, and expression analysis of PUB in cotton. The research was carried out as follows steps. First, specific species of cotton and Nicotina pentamina seeds were selected from Cotton Adversity Research Laboratory at Chinese Academy of Agricultural Science. The seeds are then planted and given cold treatment in 5 degrees Celsius. Next, the plant leaf was harvested and frozen with liquid nitrogen for use. Cotton genome data for species Gossypium rainbowi, Gossypium arboreum, Gossypium hirsutum, and Gossypium barbadans were obtained from cotton gene. U-box domain profile was obtained from PFAM and used as a query to identify the candidate of PUB genes from cotton genome protein database. After that, BLAST was used to obtain the coding domain sequence, protein sequence, and corresponding full-length sequence in genome. Protein sequences are further analyzed in SMART and PFAM to ensure that each candidate of protein contains a U-box domain. Moreover, CELLO is used to predict subcellular localization of proteins. Next, gene structure are analyzed by using coding domain sequence and genome sequence with software GSDS. Full-length sequence of PUB proteins were used to construct a phylogenetic tree using cluster X and neighbor genie method. The distribution of cotton PUB gene on chromosome generated with MapInspect. Other than that, homologous gene pair were identified according to the result from multiple sequence alignment. Gene GH-PUB85A and gene GH-PUB45D were selected for expression. The genes were cloned into vector, and after that, transformation, amplification, and selection were carried out. Infusion primers were then designed based on the vector sequence to construct silencing vector, RFP vector, and plant overexpression vector. These vectors were transformed into cotton and tobacco with agrobacterium-mediated transformation method, and the gene expression were observed. After analysis, 93, 96, 185, and 208 PUB genes were found in D5, A2, A1, and A2 genome respectively. Tetraploid cotton species were found to have twice PUB genes than in diploid, showing that PUB genes were relatively conservative in evolution. The amino acid content in U-box domain is almost identical, which is 75 amino acids. In subcellular analysis, the researchers were found that PUB protein distribute throughout the cell, especially in the nucleus. Generally, the evolutionary relationship is correlated with gene structure. That is, exons with more similarities in terms of size and number have closer evolutionary relationship. Exon members for 82 were much different compared to other 3 species, and due to this, only the later 3 species were used in evolution relationship analysis. For chromosomal localization analysis of PUB gene, they found out that most of the genes are on chromosome and only some on scaffolds. The distribution of PUB genes are uneven in diplody D5 and A2. In Gossypian hirsutum, which is tetraploidy, PUB genes are located most on chromosome D07 and least on D08, compared to the other chromosome in A and D subgenome, proving that these two chromosomes were relatively conserved in evolution. In Gossypian barbadans, which is also a tetraploidy, the PUB genes are equally distributed in both subgenomes but uneven in chromosome. This might be correlated with differentiation of progenitors. In gene duplication analysis, homologous pairs of PUB genes were found uneven distributed on chromosome, which might be caused by gene duplication or partial fragment replication in evolutionary history of katan. Based on multiple sequence alignment, 18 and 27 homologous gene pairs were discovered in D5 and AD1 respectively. Most of the homologous gene pairs are due to segmental duplication and only few of them are tandem duplication. The homologous gene pair of PUB gene between D5 and AD1 were 197 pairs. Out of that, 116 pairs were located in subgenome D. On the other hand, homologous gene pair between AD2 and AD1 were 191 pairs. Out of that, 106 were located in subgenome A. This indicates that more than half of the homologous genes in G. hirsutum AD1 were derived from corresponding cotton genome. Next, the expression of PUB genes were found differently in different parts of plant. In this experiment, GH-PUB85A and GH-PUB45D from subgroup 3 were cloned and studied. They found out that both genes has high expression after 6 hours under draft stress and 12 hours under salt and cold stress. This indicated that the responses of the genes towards draft stress is much faster than in salt and cold stress. As a conclusion, this study has provided insight into response mechanism to abiotic stress. The PUB gene in evolution were highly conserved but significantly different in terms of gene structure due to tandem and segmental duplication. GH-PUB85A and GH-PUB45D were cloned and found that they were responded to abiotic stress positively but contribute differently. The genetic information from this study could provide some invaluable information for functional analysis of PUB in katan in the future. Thank you.